Arrestins are a small family of proteins important for regulating signal transduction. Function: arrestins were first discovered as a part of a conserved two-step mechanism for regulating the activity of G-protein-coupled receptors in the visual rhodopsin system by Hermann K1-Hn and co-workers, and in the I-squared adrenergic system by Martin J. Lochs and co-workers. In response to a stimulus, GPCRs activate heterotrimeric G-proteins. In order to turn off this response, or adapt to a persistent stimulus, active receptors need to be desensitized. The first step is phosphorylation by a class of serine 309 kinases called G-protein-coupled receptor kinases. GRK phosphorylation specifically prepares the activated receptor for arrestin binding. Arrestin binding to the receptor blocks further G-protein-mediated signaling and targets receptors for internalization, and redirects signaling to alternative G-protein-independent pathways, such as I-squared arrestin signaling. In addition to GPCRs, arrestins bind to other classes of cell surface receptors and a variety of other signaling proteins. Subtypes: mammals express 4 arrestin subtypes, and each arrestin subtype is known by multiple aliases. The systematic arrestin name plus the most widely used aliases for each arrestin subtype are listed in bold below. Arrestin 1 was originally identified as the S antigen causing uveitis, then independently described as a 48K DAR protein that binds light-activated phosphorylated rhodopsin before it became clear that both are one and the same. It was later renamed visual arrestin, but when another cone-specific visual subtype was cloned, the term rod arrestin was coined. This also turned out to be a misnomer. Arrestin 1 expresses at comparable very high levels in both rod and cone photoreceptor cells. Arrestin 2 was the first non-visual arrestin cloned. It was first named I-squared arrestin simply because between two GPCRs available in purified form at the time, rhodopsin and I-squared-2 adrenergic receptor, it showed preference for the latter. Arrestin 3. The second non-visual arrestin cloned was first termed I-squared arrestin 2, even though by that time it was clear that non-visual arrestins interact with hundreds of different GPCRs, not just with I-squared-2 adrenergic receptor. Systematic names, arrestin 2 and arrestin 3, respectively, were proposed soon after that. Arrestin 4 was cloned by two groups and termed cone arrestin, after photoreceptor type that expresses it, and X arrestin, after the chromosome where its gene resides. In the Hugo database its gene is called arrestin 3. Fish and other vertebrates appear to have only 3 arrestins, no equivalent of arrestin 2, which is the most abundant non-visual subtype in mammals, was cloned so far. The protochordate C. intestinalis has only one arrestin, which serves as visual in its mobile larva with highly developed eyes, and becomes generic non-visual in the blind sessile adult. Conserved positions of multiple introns in its gene and those of our arrestin subtypes suggest that they all evolved from this ancestral arrestin. Lower invertebrates, such as roundworm C. elegans, also have only one arrestin. Insects have A1 and A2, originally termed a visual arrestins a because they are expressed in photoreceptors, and one non-visual subtype. Later A1 and A2 were found to play an important role in olfactory neurons and renamed O sensory. Fungi have distant arrestin relatives involved in pH sensing. Tissue distribution: one or more arrestin is expressed in virtually every eukaryotic cell. In mammals, arrestin 1 and arrestin 4 are largely confined to photoreceptors, whereas arrestin 2 and arrestin 3 are ubiquitous. Neurons have the highest expression level of both non-visual subtypes. In neuronal precursors both are expressed at comparable levels, whereas in mature neurons arrestin 2 is present at 10 to 20-fold higher levels than arrestin 3. Mechanism: arrestins block GPCR coupling to G proteins via 2 mechanisms. First, arrestin binding to the cytoplasmic tip of the receptor occludes the binding site for the heterotrimeric G protein, preventing its activation. Second, arrestins link the receptor to elements of the internalization machinery, clathrin and clathrin adapter April 2, which promotes receptor internalization via coated pits and subsequent transport to internal compartments, called endosomes. Subsequently, the receptor could be either directed to degradation compartments or recycled back to the plasma membrane where it can once more act as a signal. The strength of arrestin receptor interaction plays a role in this choice. Tighter complexes tend to increase the probability of receptor degradation, whereas more transient complexes favor recycling, although this only is far from absolute. Structure: arrestins are elongated molecules, in which several intramolecular interactions hold the relative orientation of the two domains. In unstimulated cell, arrestins are localized in the cytoplasm in this basal oinactive via conformation. Active phosphorylated GPCRs recruit arrestin to the plasma membrane. Receptor binding induces a global conformational change that involves the movement of the two arrestin domains and the release of his C-terminal tail that contains clathrin and APRIL2 binding sites. Increased accessibility of these sites in receptor-bound arrestin targets the arrestin receptor complex to the coated pit. Arrestins also bind microtubules, where they assume yet another conformation, different from both free and receptor-bound form. Microtubule-bound arrestins recruit certain proteins to the cytoskeleton, which affects their activity and/or redirects it to microtubule-associated proteins. Arrestins shuttle between cell nucleus and cytoplasm. Their nuclear functions are not fully understood, but it was shown that all four mammalian arrestin subtypes remove some of their partners, such as protein kinase JNK3 or the ubiquitin ligase MDM2, from the nucleus. Arrestins also modify gene expression by enhancing transcription of certain genes. References. External links: Arrestin at the U.S. National Library of Medicine. Medical subject headings.